This is a presentation on genetically modified organisms. On June 10, 2011, an article was published on spider silk spun by silkworms. There are many scientists working on this project, the most cited being Mr. Donald Jarvis. Here's how it works. There are certain proteins that make the silk woven by spiders for their webs especially strong. For its size, it's even as strong as steel. The scientists working on this project saw potential and mass production of spider silk. Scientists had manipulated cells and even goats before to create this substance, but an importance lies in its spinning. In order to solve this problem, the scientists decided to attempt to make a silkworm create spider silk. The worm silk, which is used to create its fluffy cocoon, is known for its sleek texture, often used in cloth, but it has little strength compared to the silk of spiders. So how do the two combine? Not like this. First, a cell is taken from the spider. The scientists could easily locate the DNA inside the nucleus of the cell. They knew what the gene was that created the protein which made the spider silk stronger. They were able to isolate this gene using a technology called piggyback. The gene coding for the desired protein is cut by transposase after it recognizes inverted terminal repeats, which are specific non-coding sequences of DNA. This section of spider DNA is pasted into a vector, which is the circular form of DNA, seen here in black. Once transported to the silkworm, transposase then cuts the spider DNA from the vector and pastes it onto the DNA of the silkworm, where the base pair sequence reads TTAA. The transposase has little limitation and can be used more than once. One cell in the silkworm then has the gene to make the strengthening protein. This cell will eventually divide, and the altered cell will have replicas throughout the organism. Piggyback transposition technology does not leave a footprint on the DNA. And after all that, the silk only has 5% spider proteins, but is about 48% stronger than regular silk. So what are the potential applications of this technology? There are many potential applications for this specific discovery. Because the silk is strong but light, it might have medical applications such as better sutures and may play a role in artificial tendons. Outside of the medical field, spiderworm silk has potential to make an even safer bulletproof vest. Since the piggyback transposon system is relatively new, there's no clear drawback. In terms of genetically modified organisms, many people believe that we should not alter the directions given by DNA, in animals or humans. It is clear interest in this area is growing, and people are afraid of potential outcomes. Others feel it is necessary to create an ethical code for this business before moving forward. As this technology grows, it will affect what DNA is passed from organism to offspring, and therefore will alter evolution.